Hello, welcome to 13 Universal Learning Beyond Barriers, and this is Shedro with you on A-Level CIE Biology. We're on A2 topics, all topics paper 4, series 8. We have done 1, 2, 3, 4, 5, 6, 7. So this is the 8th one. Please make sure you watch these videos, different topics treated. Okay? So get your writing materials because it's time to solve questions. Remember, what we do in this video is to solve questions. All right. So come, let's try your concept. Guys, remember the rule of engagement. Do not check the math scheme before solving this question. Do not check the solution. All right. So just work with me. Get your materials. Let's do this. Okay. So the first question says an array comparative genome hybridization (ACGH) is a technique involving the use of a microarray to analyze a genome or sections of a genome. Outline means they should give the main features. Outline the steps required to prepare the genome of an individual so that the genome is ready for analysis using a microarray chip. So pause this video and try to retrieve. Remember that most important thing we do in biology, this is a structural question, we practice retrieval. Okay, being able to retrieve, remember, okay, what you have learned. Memory is key in biology. I keep saying it. We do not just want you to understand concept, we want you to remember concept because you will be tested. Right, so this is what is going to happen now. Pause this video and try to remember the steps required. Okay, put them in a sequence. Welcome back. So the first step required in microarray is that first of all, extract or isolate your DNA. Okay, you have to isolate or extract your DNA from cells. All right, and once you extract your DNA from cells, then you can amplify it if it is small using PCR. You can amplify it if it is small using PCR. Okay, then of course, if it is okay, then you go to the next step. All right, the next step is that you have to cut DNA using restriction enzymes. Cut them into fragments using restriction enzymes, and then you have to produce single strands by denaturing the DNA. DNA is double-stranded, so by the time you've cut them into strands, remember that they are joined together by hydrogen bond between the complementary bases. So you have to denature these DNA to have single strands. Okay, because what we need is single strand so they can bind with the probe in the microarray. Okay, all right, so now you've denatured. The next thing you have to do is a label. Okay, tag the DNA, all right, by mixing it with fluorescent dye. Okay, and all of that, and that's all you need for this question. Okay, and that's 3 marks. Right, so let's look at what the math scheme says. Okay, any three from the following. So isolate, extract DNA from cells, tissue name, whatever name you call it. Then to cut DNA into small fragments. So you cut into fragments using restriction enzymes, and then you denature into single-stranded DNA, and then add fluorescent or tag marker, and then you're good to go. All right, the next question, please. Now, this is another question. In paper 4 and biology generally, we give you bulky, all right, context, background information that has a lot of new stuff and unfamiliar, all right, material. But you have to read it, and each line you read matters a lot. Read it immersively. Read it to understand it and remember it, because you're going to need clues. Your answers, all right, the responses will need clues from all of these big essays. Okay, let's go. The George syndrome. The George, that's the name of a person. I think this is a king, one of the kings of England. Okay, King George. I don't know, but just find out. The George syndrome is a dominant inherited disease in humans. The George syndrome is caused, all right, so number one is a dominant. So pay attention to some of these keywords. You're going to need them. Number one is a dominant inherited disease in humans. Okay, the George syndrome is caused by deletion of a large number of nucleotides from chromosome 22. So pay attention to this. Large number of nucleotides deleted from chromosome 22. The next paragraph. The number of nucleotides deleted varies between individuals. Wow, it varies between individuals. Okay, now if it varies between the individuals, that means I can have two people that have the George, all right, but you know they will have different nucleotide, all right, deleted, right? So that means that this will have different alleles, because look at what it says. The number of nucleotide deleted varies between individuals. So as you are reading, you are thinking. As you're reading, you are thinking. All right, that's a massive reading. You immerse yourself into this. So the number of nucleotide deleted varies between individuals in a range from 800,000. Wow, that's too much. To 3,100,000. Wow. So that means somebody that has the the nucleotide deleted 800, right, may not be this exact, may not have exactly the same phenotypes, have the same phenotype as someone that has 3,100,000 deleted, right? Because the more nucleotide deleted, the more genes deleted. That means the person that has 800 deleted will have more genes compared to somebody that has 3,100,000 deleted. Okay, so they're going to give more, right? Different, they'll be different in phenotype. Just as you are reading, you are thinking. The largest deletions can cause removal of up to 46 protein coding genes from the chromosome. So the more deletions, the more genes removed. So the more characteristics lost. All right, more proteins cannot be made. Okay, next paragraph. Fig 4.1 shows the result of ACGH using a microarray specific for the section of chromosome 22, within which, okay, the George syndrome deletions occur. All right, so this figure, we're going to look at the figure. It results ACH using a microarray, okay, section of DNA within which. So we want to find out where this has occurred. That's what I want to do. So you slow down and understand it. That's how to read this stuff, okay, so that you understand what you're reading. Don't just read on the surface. Read in between the line. Think through. Every information matters. The microarray analyzed DNA from two individuals. How did they analyze it? One with the George syndrome. So two people, one with the George syndrome, the other one who did not have the George syndrome. That's the control DNA, the one that doesn't have it. Okay, so the genes are not deleted. Okay, so some genes are missing here, genes are intact here. In the ACH results shown in Fig 4.1, we see the figure 4.1. Each small circles represent the result from a single probe on the microarray. The x-axis shows the position of each probe on chromosome 22. The position is shown as distance along the chromosomes in millions of nucleotides. That's x. As a result close to 100%, right? Fluorescence on the y-axis means that the DNA from the individual with the George syndrome fluoresces at the same intensity as the control DNA for that probe. Okay, yeah, so they have the same genes, right? But as a result, close to 50% fluorescent on y-axis means that the DNA from individual with the George syndrome fluoresces half as such as control. Okay, what does this mean to you? What it means, because as you read, you ask yourself the meaning. What this means is that in this 50% fluorescent, for this 50, that means this is where some genes are actually missing, right? Some genes that are not found in this individual with the George syndrome, but are found in the genome, right? Okay, because shown by the control DNA. So as you read, you ask meaning. Now let's go on. Now we have read this before, okay? So you can read it again. So let's go to the question. With references to Fig 4.1, estimate the number of nucleotides deleted from affected chromosomes 22 in the individual with the George syndrome. Give your answer to 100,000, okay, nucleotide. Let's see, guys. See this. Now remember that they already told us here that x-axis shows, right, the position on the chromosome. Look at the position on chromosome 22, and y-axis shows us, of course, the percentage, right, of fluorescence, right? Well, okay, yeah. So now, so anywhere there's 100, that means that on the y-axis from individuals with the George syndrome, fluorescence at the same intensity. But anyway, that's 50. And the question is asking with reference to Fig, estimate the number of nucleotides deleted. Now, that means where is deleted is where there's 50. 50 is only for the George, so go to 50. All right, so watch this. This is in millions. 16 to 17 is one unit, and that 1 unit represents 10 boxes. So one box here will be 1 divided by 10, which is 0.1. Okay, so let's see. All right, here this is 17, 17.1, 0.2, 0.3, 17.3, right? Yeah. So if you come here, let's look at this. This is 20. If you go backwards, 20.9, 20.8, 20.7. Sorry, 19. Because we're going backward. So this is going to be 19.9, 19.8, 19.7. So that's 17.3, 19.7. Okay, so minus it. 19.7. Okay, 19.7. Get your calculator. All right, 19.7. Want to find, all right, estimate the number of nucleotides deleted. 19.7, very important, minus, okay, minus 17.4. Guys, yeah, 1 2 1 2, okay, 17.3. Yeah, so what does that give you? That gives you 2.4, right? Yeah, 2.4. Yeah, that's 2.4. Pardon my listen, that's 2.4. Let me delete this. That's 2.4. This is 2.4. Now 2.4, but that 2.4 need to be converted to millions. So times 1 million. Okay, 1 million is 10 raised to power 10 raised to power 6. That will give you 2,400,000, right? 2 million, because they said we should leave it in the nearest 100,000. So 2 million, okay, 400,000. That's the number of nucleotides deleted. Okay, yeah. So in the judge DNA, right? So how did I calculate it? 50% is the judge, all right? They said it. Percent on the y-axis means that DNA from individual with the judge. So that means this is where the genes are lost, right? So check from here to here. Okay, you have to know the value of this, and then this is 19.7. That this point is 17.3 minus, so you can get from here to here. That convert to millions, because this is in millions. All right, let's go to the next question, guys. So of course, there's a fluorescence of DNA from an individual with the judge syndrome as a percentage of fluorescence of control. That's what we're doing here. Yeah, so let's look at that. That's the answer here. Okay. All right, let's go to the next question, guys, quickly. Explain how the microarray technique works to give the result shown. How does it work? Okay, remember, guys, you should pause this video and work this thing out. Every question, anytime you get any question, try to do it. All right, taking cognizance of what you have read, the topic, what you know about the topic, and the command word. Okay, explain means give reason, connect it to the knowledge, the concept of biology on this topic. Explain, give reason. Okay, why? Right now, so how does this work? Explain how the microarray techniques works to give the results shown. Okay, remember that the first thing you do is that when you have extracted the DNA of the person with the judge, okay, and the one that is serving as control experiment, so what you do is that, so you continue to write that the DNA of the person with the judge syndrome and the DNA of the person without the judge syndrome, they are labeled with different fluorescent tag, okay, to identify them. So the next thing they have, because they said how, so there's a sequence there. So then the DNA fragments are then mixed with the DNA probes in the microarray, and then hybridization is allowed to occur. Hybridization is when these DNA strands form hydrogen bonds with the probe, complementary probe. Okay, then it's allowed to occur. When it's allowed to happen, because how did it get the result? The next thing you write, of course, you can wash off the ones that did not hybridize, right? But what you see is that it is now, okay, viewed using UV light. UV light is shine upon microarray or laser scanner. So you find out that, all right, the region where there's 100%. Now, of course, you know that you know, we're going to have less DNA from the judge binding, hybridizing, because there are fewer genes present, okay, compared to that of the control. But the region where there's, you know, high light intensity, that's 100%, okay, means that, all right, that is where we have genes are not deleted. We have both the judge DNA fragment and that of the control, right, binding to the probe, but where we have less, okay, intensity or fluorescence, that means that that's the judge, okay. The judge, all right, that shows where the judge does not have nucleotide. Those genes are missing from the person with the judge experiment. So see it here. So DNA from the judge syndrome, and they're labeled with different color, okay, for identification. So DNA hybridizes. So anytime you are dealing with microarray, there must be hybridization on microarray. The microarray contains the probes. So the George syndrome, okay. So the George syndrome, less DNA binds to the probe because some genes are missing. So there's less 50%, okay, fluorescence when nucleotides have been deleted. Okay, fluorescence is equal 100% when nucleotides have not been deleted. So where they've been deleted, it'll be 50%, or 100%. But this is how it works. So you must label them, and you must allow them to hybridize. And then when you hybridize, you now use, all right, the information given. So of course, less DNA will bind to. So the one that has less, okay, 50%, that means genes have been deleted for this guy, the judge. So the one that have 100% genes have not been deleted. Okay, all right, just 3 marks. Suggest why the phenotypes of two individuals with the George syndrome can be different. Oh boy. Mistakenly clicked the answer. All right, don't worry. So suggest why. Let me explain it. Now, why will it be different? Of course, you've seen what the mask scheme says, but why? Remember that phenotype here, okay? Anytime they ask you why phenotype is different, remember this formula, that variation in phenotype is equal to variation in genotype, all right, plus variation in environmental factors, that's VE, okay. So don't forget that the phenotype of in two individuals with the George syndrome will be different. Remember, they told us that the number of allele deleted can be from 800,000, right? 800,000, okay? 800,000 to about 3.1 million. Did you get? Yeah, 3.1 mil. So it's going to be different because different alleles are deleted. Do you understand? All right, so they have different, all right, different alleles. So different nucleotides are deleted. Yeah, different nucleotides. So somebody that has an individual that has only 800 deleted will have all right, less nucleotide deleted. Of course, this person will have less genes, all right, deleted compared to somebody that has 3.5, 3.1. So because this has less genes deleted, so it will produce more proteins than the person with this, okay, or the person with 3 million, or the person with 2 million. You get? So they will have different alleles, all right. That talks about difference in genotype, okay, because different nucleotides are deleted, okay. Yeah, so then therefore different proteins will be made by these two people, right? And of course, you also have to mention the fact that, right, then environment also could be a factor. So it depends on what they're exposed to, the diet, all right, and of course the environmental condition, because environment influences variation, right? But don't forget that that's very important. Different number of nucleotides may be deleted. Yeah, we'll say that. Different genes may be deleted. Yes, because there's a range. Okay, an individual has only one of deleted genes, so you always have different individuals may have different alleles of the same gene, or in one individual remaining allele may be recessive but the other one are dominant. Okay, yeah, it can be if the number deleted is less than 800,000, may be recessive. Okay, if it's high within this range, it's going to be dominant. So different proteins, because if different genes, all right, are deleted, different proteins will be made because genes code for proteins. Reference to individuals Okay, maybe different environment like diet. Okay, so remember, you cannot only solve this question if you follow the background information very well. Okay, and anytime they say you should suggest, that means you are going to think outside the box, but based on what you've been given, what you know. Mhm. Always go back to the background information. They are always clues for you. All right, meiosis described as a reduction division because the number of chromosomes in the daughter cells is reduced by half. Okay, table 5.1 describes some of the events that take place during 4 of the different stages of meiosis in an animal cell. Let's do it. So this is it. Sorry, this is very time. This is metaphase. Okay, so what is happening in metaphase? Attach. All right. This is attach centromere and arrange homologous pairs of chromosomes at the equator of the cell. So this is anaphase. So let's see what they want us to do, guys. There's a complete table 5.1 by outlining the behavior of the spindle fiber during anaphase 1. Okay, identifying the stage of meiosis in which the spindle fibers reform. So identifying the stage of meiosis, that's what you have to, in which the spindle fibers reform. Okay, the spindle in the daughter cells, drawing a diagram to show. So look at this. Okay, this is spindle fiber. So what is happening in anaphase 1? Can you see what is happening here? So you see that the spindles, they shorten at the pole and they contract. They shorten and contract at the pole, and then they now pull. Now what they are pulling is in whole chromosomes, okay, apart. So homologous pairs are pulled apart, whole chromosomes, not chromatids. All right. Okay. So it is in anaphase 2 or mitotic anaphase where centromere has to split and you put sister chromatids apart. But here, anaphase 1, what you are pulling apart, guys, is whole chromosomes. Okay? Whole chromosomes, something like this. If I have this homologous chromosome, right? This is homologous chromosome here. Look at this. This is a sister chromatid of this chromosome. But this chromosome, right, is kind of attached to another chromosome, so they are both bivalent, right? Yeah, so they are bivalent. So during anaphase 1, what is happening is that the two chromosomes are separated. Okay, so this is pulled to this part of the pole. This is pulled, and how it is pulled is because sister chromatids, spindle fibers contract, they shorten, they depolymerize, and then shorten at the poles, pulling these whole chromosomes, all right, of bivalent apart. Okay, but you know the if it is in anaphase 2, in anaphase 2, what is actually separating is sister chromatids. Anaphase 2 and mitotic anaphase, what separate there is sister chromatids because centromere split. So the difference between them is that number one, in anaphase 1, what separates is whole chromosomes, okay, while anaphase 2, what separates is sister chromatids, and here in anaphase 2, centromere splits, but here it doesn't split. Okay, all right. Now where does it reform? Reform spindle in daughter cells. So spindles are reforming, they are reformed. Spindle formation begins in prophase. So this should be prophase 2. Okay, after this prophase 2, anaphase 1 jumps to prophase 2, then telophase. Of course, telophase 2, they are disassemble. So they want you to draw the diagram. So the diagram, you draw 4 daughter cells. Of course, you will have to draw 4 daughter cells there. Okay, 4 daughter cells. Yeah, 4 daughter cells. So sister chromatids. Of course, we have single sister chromatids in each of them, but spindle fiber has already disintegrated here. So, and of course, nuclear membrane reforms. Let me just join it. Okay, and then you have a sister chromatid here like this. Okay, so nuclear membrane also reforms here, right? So you have a sister chromatid here. Okay, now spindle has already, okay, all right, degenerated. So yeah, so yeah, but you see what I've drawn here is when cytokinesis has occurred. So see what you're supposed to do. Don't worry, let me finish. You have to draw it as telophase. Okay, so what I'm supposed to do here, look at this. Remove this, remove this. Okay, then you see, join these two together. Okay, so sister is the cytokinesis has started. Okay, beginning to form. Can you see? Yeah. So you see the spindle disintegrating. Okay, can you see this is disintegrating here? All right, so look at the similar thing is done here. Sorry. Yeah. Okay, so because this animal cell, so cleavage furrow starts forming. That's invagination of cell membrane. So you can see spindle start disintegrating. All right, but you see what we have here is sister chromatids, okay, at the poles, and of course nuclear membrane reforms. Sorry, let me remove this here, remove this. Yeah. Okay, see spindle reforms. Okay, that's it, guys. All right, so let's see what the mass scheme says. Make sure you are solving these questions. All right. Yeah, that's what the mass scheme says. Yeah. Yep. So look at it. So you can see that. So if you check here, you can see this is contract pull centromeres or chromosomes towards that. You can never see pull chromatids because these chromosomes are separating. Okay, towards the opposite pole to the central. So you can see that's prophase 2. Okay, 4 daughter cells, 2 single chromosomes inside, reforming nuclear envelope. All right, so reforming. Okay, so let's go to the next question. 4 marks is that. Explain the need for a reduction division. Why do we need a reduction division? Okay, during meiosis. Explain the need. Explain. Give us reason. What is the need for reduction division? Okay, the need is to form haploid gametes. Why do we have them? Okay, haploid gametes. It is 2n. Okay, so this is a cell to form haploid gametes. So this is going to give us a haploid cell, which is gamete. So the need is for formation of gametes. Remember that meiosis occurs during gamete formation. So to form gametes, right? The gametes are haploid, right? Of course, you know this is going to also divide again to give us, there are 2 mitotic divisions, right? So it's going to give us something like this. Okay, all right, I have something like this. So having 4 haploid cells, and they're all gametes, sex gametes. Okay, yeah, so 4 haploid, so that's 2 divisions. Okay, now what is the need for it? So if I have this person undergoes meiosis and forms a haploid gamete, which is N here. Okay, so haploid gamete N, right? That's N. And I have another person in their reproductive organs, okay, undergoing meiosis, and they have, so now the first need is to form, all right, gametes which are haploid, all right, so that at fertilization the gametes can fuse and produce, all right, a diploid zygote to end. See, a diploid zygote. So the diploid zygote now contains the both chromosome, both the maternal chromosome and the paternal chromosome. They are now in this zygote. Okay, and why we have to have them reduce it is to prevent doubling of chromosome number. If you do not have it, you doubling of chromosome number will occur. Imagine if you do not have this, and we're having 2n as gamete, right? And we're having these 2n, maybe meiosis did not occur, 40 meiosis occurs, all right, the chromosomes refuse to separate, So our gamete is, instead of having haploid, the gamete is having diploid. All right. So by the time they fertilize, we're having a tetraploid. All right. So this is dangerous, it's scary, because the offspring here will have double right of the parental features characteristics. If he has 2 eyes, this will have 4 eyes, double of 2. Okay, 1 head, 4 head, you know that. So reduction prevents doubling of chromosome number, okay, which is what we call polyploidy. That's chromosomal. Okay, look at it. Any for meiosis, okay, produces gametes. You can see 2 gametes fuse at fertilization to form a zygote. Zygote will have maternal and paternal chromosomes. Gametes, right, haploid. So the zygote is diploid. Yes, prevent doubling of chromosome number. So that chromosome number remains constant every generation. Okay, doesn't double. Okay, let's go to the next question, guys. Fig 6.1 is a diagram of a section through a mitochondrion. The 4 arrows show A, B, C, and D. So the answer says show the movement of molecules and ions, right? Active transport of protons. So active transport of protons. Remember that protons are usually pumped from the matrix into the intermembrane space. Okay, active transport. All right, pumped from, that's it here. Okay, during electron transport chain, the energy release is used to pump proton from here into the intermembrane space, so that proton gradient is set up here. Okay, all right. So this is D, that shows active transport of proton. Then diffusion of carbon dioxide. Carbon dioxide is produced during link, of course, link reaction and Krebs cycle. They can move from matrix to here and diffuse out. So D and C, they're just moving out. You can say C and D. Okay. All right. So let's look at the next question. Explain how a lack of oxygen affects oxidative phosphorylation. Right. So if oxygen is not present, what happens? So the first question you ask yourself is, what is the role of oxygen? Okay. So pause the video and try to solve it. Oxygen is a final electron acceptor. Remember that oxygen is a final electron acceptor. If you remember the electron transport chain, so oxygen is the final electron acceptor. So remember, during electron transport, this is the first carrier. Okay, passes the electron to the second carrier, here, right? Passes the electron to the third carrier. All right. Okay, this passes it. Let's assume that there were 4th carrier, and then yes, 4th carrier, and let's assume, of course, now the final electron acceptor is always oxygen. So oxygen is the final electron acceptor, so it combines with electrons. So you see electrons move from one carrier to another down, okay, energy level. This electron moving from C1 to C2, right? So from C2, right, electron moves from C2, right, to C3. All right, so that's the redox reaction. Loss of electron is reduction, gain is oxidation. Okay, gain of electron is reduction, gain, all right, loss is oxidation. All right, so you can see that. So this combines with proton. It combines with proton. So oxygen is the final electron acceptor. So it accepts this electron with proton, okay, to form what? Water. This is how water is formed. So this is how water is formed here. Okay, this is how water is formed. All right, so water is formed here. Can you see? Yeah. So oxygen is the final electron acceptor. Okay, so remember that NADH, all right, NADH2, like, and of course, what happens there is that NADH2 and FADH2, or reduced NADH, when they arrive here, they undergo oxidation. Okay, they lose their hydrogen to regenerate NAD. Okay, All right, so NAD is formed here. That's NAD. Okay, so this oxygen, this hydrogen, they release the hydrogen, right, it then, okay, is donated to the electron carriers, these guys. And the electron carriers, what they do with hydrogen is that they split hydrogen into proton and electron. So proton and electron, so this is proton and that's electron, remember? So just trying to revise this. Okay, okay, you can put to here to electron. So electron then enters electron transport chain, moving from one carrier to another. Okay, so now remember that the energy, when electron moves down energy gradient, right, the energy release is used to set up proton gradient in intermembrane space. Okay, so the question says explain how lack of oxygen affects. So if there's no oxygen here, there's no electron acceptor, fine electron acceptor. Okay, so what is not formed, so that means that the flow of electron will stop, electron transport chain will stop. Okay, right, NAD will not be regenerated. Okay, and of course chemiosmosis will not occur, right? So less energy will be formed, oxidative phosphorylation will not occur. And if NAD and FAD they are not regenerated, what happens is that Krebs cycle link reaction will shut down. Do you get? All right, let's see what they say here. Look at it. Na3 site of link reaction, Krebs cycle. Okay, then okay, explain how local oxygen of, okay, this is a different answer to this. It's a different answer. Okay, but you understand what I said, right? That if it's a different answer, is a mistake, right? If oxygen is not present, so as how a lack of oxygen affects oxidative phosphorylation, it will not occur. Electron transport chain will not occur, right? Remember the oxidative phosphorylation, guys. Oxidative phosphorylation is equal to electron transport chain, etc. All right, and what? Chemiosmosis. So electron transport chain powers chemiosmosis. The energy released by electron is used to set up electron and proton gradients. So that's chemiosmosis. Right? Chemiosmosis. Chemiosmosis. Osmosis. All right. So if oxygen is not there, no final electron acceptor, no electron transport chain occurs. Of course, oxidative solution will not occur. Chemiosmosis will not, any ATP will not be made. Okay, and of course NAD will not be regenerated. So all of those link reaction and Krebs cycle shut down. Okay, no proton gradient setup. All right, so that's what we stop today. So thank you. Share your comment with us. If you have any need, let us know. Share this video. See you next episode. Bye. This is Shedro.